There's also a lot of diversity in flower color and plant architecture, which a lot of us outside this room aren't really aware of. Next slide. So cultivar definitions really monitor this diversity. The interesting thing about cultivars is a cultivar is a plant that's defined by its interaction with its humans. So cultivars are pulled out from populations through selection by their human caretakers, right? So cultivars aren't wild plants. By their definition, they are maintained by humans, and their phenotype that defines that cultivar is driven forward by conscious selection of the humans tending them. Okay, this is something that's very important. All of the cultivars you see up there, like roses are a great example. It's very analogous to cannabis, where you have hundreds of varieties in certain regions and thousands of varieties globally. And the definition of a rose is based on a multifaceted input. So by where you develop a new rose, you have an entire panel of roses by which you need to reference to make sure that that new creation is in fact unique. Next slide. So the term cultivar, this is like, I really don't believe in words on slides, but these are very important words. The term cultivar most commonly refers to assemblages of plants selected for desirable characteristics that are maintained during propagation. So selected and maintained are the two most important words in that definition of what a cultivar is. And these definitions are not just laissez-faire. They're codified terms in the international horticultural world, and they're very strict in how we can define our cultivars. So I'm going to take a little bit of a diatribe because we're also talking about varietals. Those words are used interchangeably a lot, but they're not quite the same thing. A varietal is a cultivar that has IP protection, right? So that's the difference. All varietals are cultivars, but not all cultivars are varietals, okay? So just a point of clarification. Next slide. So how do we go about defining cultivars? Well, I think it's a lot easier to talk about dogs. I'm a crazy dog person. So maybe this will ring true for you. But when I come to this party and I say, I have a boxer, and my mom has a Doberman, we all have a picture of what that breed looks like. And so that's where we're trying to get. When you say a name of a plant, that name is attached to something tangible that has a list of definitions that we all come to consensus on. That does not necessarily mean they're clones of one another, because plant cultivars can either be clonally propagated or seed propagated, and within groups, you can have both clonally propagated cultivars and seed propagated cultivars. The importance is that the definition of those boundaries are clearly laid out and then maintained in the propagation process. So, right, cloning is much easier. You're just perpetuating the plant, whereas seed lines are a little more complicated, yet we have them in all kinds of plants. We have both types. Next slide. So this one we're going to have to go up and down a little bit, but this is something that's really important because we're in the genomics era, and I'm a population geneticist, so I really love genetics. And I'm here to tell you genetics don't define cultivar. Okay, let's say that again. Genetics do not define cultivar. Okay, cultivars are defined by the genetics, so your genome of your plant that interacts with your environment. So everyone in this room has had a friend who's growing the same cut, and it looks totally different, right? And so we all are very, very aware of this interplay, G by E, this genetic and times environment interaction, and how that plays into phenotype. So if we slide this up slightly, we'll see that the definition of a cultivar is actually maintaining that cultivar during propagation in conjunction with the phenotype that we are defining that cultivar with, and then documenting the diversity of that cultivar across a landscape. Okay, so this should scream Appellations. This is why growing a cultivar in one location as opposed to another may actually define an appellatory accession, because it does so much better here, or it has a different quality in this particular place. And so it's a combination of those things that actually makes a cultivar. Okay, we're going to dive in a little bit more. So next slide. So this is, actually let's go up a little, just get rid of cultivar. We don't need cultivar, we're gonna go to here, yeah. So this is what I mean by genetics don't define cultivar. So this is really confusing, but you can have the same genome and have two distinct cultivars. Or you can have disparate genomes that create the same cultivar, okay? So a really good example of different genomes with the same cultivar is the fingerling banana, the ladyfinger banana, that's like the most common banana in the grocery store. There's entire populations of two Musa species. There's a lot of diversity in mom A and dad B. The genomes are totally different within those dads and totally different within those moms, but anytime you cross those two together, you get a ladyfinger banana. Right? So the genomes are different, but the cultivar itself, the type, is defined by the phenotype, starch content, shape, sugar, all these things that define that banana. So different genomes, same cultivar. On the other side, a lot of us in this room have seen this, where you have a plant, and you'll see a variegated sector. So on this plant, it's a yellow rose, and all of a sudden you have a white flower that's on the same plant. Okay, you can't get more of the same genome than that. It's the same individual. And that mutation, it doesn't matter where it is. It doesn't matter if you even know where it is. In most cases, we don't know where that mutation is. You can take a cutting of that white flower, propagate it, and you now have a new cultivar. Same genome. Right? So this is why it's really important to realize that it's the human interaction and the human definition of cultivar that drives that thing that has value. So next slide, how do you do that? It seems really complicated, right? So here, what you do is, so this is just like another way of looking at this that's so helpful for some individuals, where you can have the same genome, you have a mutation, and then you have cultivar A and B, and then same thing, you have multiple different genomes that then come together to make the same type, regardless of genotype in the background. Okay, yeah, so there's a little bit of formatting issue, but we'll get through it. Next slide. That's just my picture to make it clearer. Yeah, it's the same slide. It's just really big. Yeah, so this just, again, we got this. We can skip to the next slide. So this is how we start this process. So anytime you go out into the world as a biologist and you find a new species, whether it be a salamander or a plant, the first thing you do is make a species definition. The most critical part of this species definition is the voucher sample. It is a physical representation of this thing. Without that, you do not have a new species, right? So you have to physically define it as a tangible object, and then you layer on top of that the physical description. And if it's a plant, you're going to have a whole different grouping of data that are going to go with that sample than if it were a salamander or an insect. But this process of pressing plants and preserving them is the most fundamental way of documentation. And we have voucher specimens that go back for decades, like hundreds of years. So the type specimen for cannabis sativa was made by Linnaeus, the grandfather of this naming system. And that pressing still exists in the Karolinska Institute in Stockholm, where I am from. And you can go and you can look at that pressing that makes a definition of cannabis, right? And so as you move down into a more granular system where you're taking a species and you're dividing it into its flavors, right? It's different brothers and sisters that make your cultivars, you would then define them the same way. And so in order to do this, we have started a cannabis herbarium because there aren't any. Right? So the most fundamental resource you have to do good science doesn't exist in cannabis. It's a big deal. And it's a really big deal now because this community knows that diversity, has stewarded that diversity, and should be the community that defines that diversity. Like, this should not be out of your hands, it's fully in your hands. You know these plants. And so how do we do this? It seems like an impossible task. It's not. So go up. This is actually kind of cool. Like, there's some just more specific herbarium specimens on the bottom. You can see, like, these are quite old. This is chamomile and different things. And you will have regional accessions of these things in addition to type specimens. So if we go up, next slide. this is how we're doing it in cannabis. So we are actually going to breeders and cultivars that have been and cultivators that have been stewarding this material and taking cuttings. So you basically take a vegetative and a flowering example and then write a definition that then is attached to that physical documented unit. You keep one, the herbarium keeps one, your local herbarium chapter will keep one, and so there's repetitive information that then links this naming system to the plants themselves. Once you have that, you can layer on any kind of chemistry, any kind of genetic data you want, and that is how you move forward with understanding how that G by E interacts. So when we're talking about, name somebody, name a plant that you love. A cannabis cultivar. Haze. Okay, haze. We're in the land of haze, right? So yeah. So if we're in the land of haze, you have a type that is haze. So you can think about this like a bully breed, right? And then you have different kinds of bully breeds. You might have different kinds of hazes based on their pedigrees that have different characteristics. And you make those definitions, and a lot of people are like, oh, well, what if we disagree? You have to have a resource by which you can have arguments. So you can come to consensus so that you can make definitions that are community-based and are based on real data. Okay? So we're doing this with cannabis. All of these plants are, the data is taken in, vouchers are made, it is self-reported, and then once we have enough information, we can start making collective decisions about definitions. Okay? Next slide. So this plays into appellations, right? So once we have those cultivar definitions, we can start to understand how they play across the environment and how those environments affect the quality and or prestige of that plant within that region. And so we have this with wine in France. And next slide. This is what we're doing in this particular process. We're looking at how the variation across an environment is going to change. So if you have the same tansy grown across a whole ecological gradient, you're going to have different phenotypes emerge. And this kind of experimentation can then help us understand where things grow best. So this is a lot of work, but we've already done a lot of the work, so it's really just about documenting what we already know. So if we all are growing the same cut in 17 different areas, and we all put those farm accessions into the herbarium, then as a community we have a resource by which we can understand how haze does different here than it does in my backyard in Sonoma County. Right? And maybe understand, oh, well, if I'm growing in Sonoma County, it's going to be super short, but it's going to be real pink. Maybe I like that. Maybe that's what I want to do. Or maybe I realize the chemical content is not going to be as great as it is here, and so it's not the plant I want to grow. So from that information, you can leverage, okay, I buy a new property. I want what plants do best here. You can make really good decisions about that. Or you can go to that resource and say, these are the kind of plants I want to grow. This is the area I'm in. What's going to do best? This is the kind of information that comes out of this resource, which is documenting what you're already doing. Okay, next slide. So these are our favorite chemicals. So, of course, you can't do chemistry on everything, but we have a lot of chemical data that we already collect. And so by asking farmers to then link their chemical data to their samples, we as a community can start to understand and unravel what drives differential chemistry production, right? Like, why is my plant full of pinene, and when my neighbor grows it down the hill in the cold valley, it doesn't smell like pine trees or lemon at all, right? Next slide. So this is how we're facilitating this process. So Canndor is really the container. It's a community-run resource whereby you make voucher samples. They're pressed. Of course, these plants are just saved for prosperity. They're digitized. Any information you want is layered on top. And the community that is contributing to the herbarium has access. You can't buy in. You got to be a member. You got to be part of the community doing this work to have access to the work itself. It's a community-run process. And I think the next slide is just from the slide. Like, where do we go? So, questions, yes?
Is there a formatted size and particular data you need for a submission?
Yeah, so the classic herbarium size is 14 by 11, but anytime you have really large plants,